Subway content dia lebih kira kepada ni ATP. Alright, so alhamdulillah we are now on live recording. Alright, so without further ado kita akan mula. So should you guys have any question ataupun nak tanya soalan, boleh just raise for my attention at any time. Okay, so Bismillahirrahmanirrahim. Okay, so here we are on new chapter. Okay, not new chapter of your life, new chapter of biology, chapter 5, cellular respiration. So as the name suggests, cellular respiration, apa yang kita dah tahu sebelum ni adalah respiration. Respiration is the process. If you're talking about a human respirates, we know they consume oxygen. I mean, they inhale oxygen, then they exhale oxygen. But now we want to discuss at cellular level. Mana how each of the cell do respirates. Okay. So that is cellular respiration. Okay. So here are the learning outcomes. Okay, subtopics in this chapter. All right. So yes, can go through this from the syllabus. All right. So I will begin with this. How do living organisms get energy? Of course, we know we get energy from the food, i.e., mostly is the carbohydrate, and then through the digestion by enzyme, we do obtain glucose, and from glucose, iaitu gula tu, okay, the cellular respiration begins to produce energy. Okay, yeah, somehow glucose is very important. Gula is important, but in excessive amount, it is not good. Tahulah akan boleh leads to diabetes and etc. Okay. Orang yang selalu diet tu dia tahulah dia try avoid glucose, try avoid this diet, that diet, intermittent fasting, blah blah, semualah avoid carbs. So yang tu you guys aware. Alright, so 5.1, importance of energy. Kita terus masuk importance of energy. Kenapa kita nak perlukan energy? Kalau dalam bahasa mudahnya memang kau dah tahulah, but scientifically why? Okay, so at the end of this lesson, you should be able to state the needs of for energy in living organism and explain ATP as the main source of chemical energy. So actually main focus dia dekat sini lah. Main focus dia pada ATP. Okay, ATP is a source of chemical energy. Okay, so it is used for metabolism. Metabolism ni refers to all of the chemical reactions that occur in the body. It means semua tindak balas kimia yang berlaku dalam badan kita panggil sebagai metabolism. So there are two types of metabolism. Okay, the word metabolism ni korang selalu dengar lah, ya, metabolism. But actually metabolism there comes in two types, whether catabolism or anabolism. Okay, both of these are regarded as metabolism. Okay, so catabolic pathways, I take the keywords only, is breaking down of complex molecule. Anabolic pathways on the other hand, building of something. Building of the complex or complicated molecule. So remember, catabolic breakdown, anabolic build. Anabolic macam ana buat, build. Alright. Energy and respiration. Okay, so untuk cellular respiration, the catabolic pathway of aerobic and anaerobic respiration, which break down the organic molecules for the production of ATP energy. Okay, so pendek ceritanya ini adalah very brief ataupun a general summary of cellular respiration. So what is cellular respiration? It is a catabolic pathway. So the term that I want to highlight here is catabolic. So if you refer from previous slide, catabolic means we are breaking down something. So what are we breaking down? We are going to break down organic compounds plus oxygen. The organic compounds is the glucose, C6H12O6. Tu kau dah hafal kan, glucose. Breakdown of organic molecule kepada production of ATP energy. Okay, so of course, as everyone knows, if you've been learning this from your secondary school, perhaps from your primary school, already have learning about this. Kat mana ATP dihasilkan? Where does ATP will be produced? Inside the organelle called mitochondria. Okay, so inside the cell. So inside the cell, there is an organelle called mitochondria. That is where you build ATP. So this is the structure of mitochondria. So it has a double membrane, ada dua membrane, outer membrane and inner membrane, bahagian dalam. So between the membrane is the space what we call as intermembrane space, ruang antara membrane. So this inner membrane dia ada infolding. Yang inner membrane ni kalau berlipat-lipat ke dalam ni namanya adalah cristae. Okay. So dia contains enzymes dan juga nak buat ATC. Sebenarnya nanti yang saya nak beritahu you guys in advance, nanti kita akan belajar macam mana ATP tu dihasilkan last sekali mostly pada membran dalam. So this membran nampak macam lipat-lipat macam lemak yang terlipat-lipat, tapi kat situ lah paling penting. Kat situ lah ATP nak dibuat. And the name of the liquid or the fluid inside this inner membrane, it is called as matrix. Bukannya matrix KMS, okay? Matrix. So it contains the DNA, ribosomes, and enzymes used for Krebs cycle. Okay. So ATP is the short form for adenosine triphosphate. Bila tri maksudnya ada tiga phosphate. We will look onto this soon. Okay, so adenosine triphosphate is the energy currency of a living cell. Kenapa dia panggil energy currency? Okay, mata wang currency. Apa benda ni? Tak faham ni sir. Okay, so what is the currency of Malaysia? Is ringgit. So there is the our commercial benda yang kita gunakan lah. So energy currency for energy adalah dalam bentuk ATP. So adalah mata wang yang digunakan untuk trade energy. Okay, dalam bentuk ATP. So the molecule from which cells derive energy. So molekul akan ambil energy daripada ATP ini. Okay, so it has the shape with which can be recognized by highly specific enzyme. So ada enzim yang boleh recognize ATP. Nanti kita akan tengok enzim apa namanya. Okay, so ATP ni contains the ribose sugar and the nitrogenous base. Ni nitrogenous base sebab tu nama adenosine sebab dia ada adenine dan dia ada ribose sugar and dia ada triphosphate. Sebab tu namanya adenosine triphosphate. Okay, the bonds between the phosphate groups of ATP can be broken by hydrolysis. So maknanya ni, bond antara one phosphate group, two phosphate groups and three phosphate groups, each of these bond can be broken down, can be hydrolyzed. Okay, can be broken by hydrolysis. So when the terminal phosphate bond is broken, terminal ni maksud yang paling hujung. Okay, by addition of water molecule, a molecule of inorganic phosphate. Kita akan sebut dia sebagai inorganic phosphate ni bila ada simbol Pi. Bila phosphate itu sudah terpisah, phosphate itu sudah terasing. Okay, the reaction is exergonic and releases 30.5 kilojoule. So maksud exergonic dekat sini adalah dia release energy ni. Bila dia break bond, dia akan release energy. Exergonic release energy. Endergonic absorb energy. You guys learn this in chemistry, right? Okay, so the reaction is exergonic. Alright. So this is what is meant by inorganic phosphate. So originally we have ATP. So bila kita break down hydrolyze this bond, so dia akan release menjadi inorganic phosphate. Maksud inorganic phosphate, this phosphate is not bonded to any carbon compound. So itulah maksudnya. Okay, so ada several slide yang saya skip. Yang tu korang boleh baca sini, selagi pun dia cuma elaboration sahaja. Okay, so now we're going to look at importance of ATP. So ATP ni function dia untuk apa? Provides energy for muscle contractions, urea synthesis, even hasilkan urea pun, maintaining a constant body temperature, active transport, Calvin cycle dan juga nitrogen cycle. Semua tu perlukan ATP. Okay, so how ATP is used in process within cells? Yep, when we do our movement even inside in cilia, flagella, the sperm beating its tail, muscle contraction, alright, that is more common to you. Movement of chromosomes during cell division, maintaining body temperature. Yep, bila kita nak perlu extra energy, okay, to provide optimum internal environment. Okay, another apart from that is anabolic process. So anabolic process ni bila kita nak build up something, anabolic build catabolic breakdown. So dia perlukan energy. So dapat energy daripada mana? Daripada ATP juga. And for active transport, okay, for certain molecules untuk transport daripada one part of the cell to another part of the cell, okay, and etc. Next, secretion. Bila kita nak transport something out into vesicles, we also need ATP. And for certain organism that do have bioluminescence ni yang macam nanti dia akan yang dalam cahaya tu dalam gelap tu dia akan bercahaya. Some organisms are able to convert chemical energy into light. Ini kadang-kadang tumbuhan ataupun haiwan ataupun ikan-ikan yang pelik-pelik dekat dalam dasar laut tu bioluminescence. Okay, so there is short and brief regarding ATP. That is what you need to know in general and a little bit detail about ATP. So you dah tahu lah ATP tu adenosine triphosphate. Dia ada 3 phosphate. Yang when you break down 1 phosphate menjadi dia inorganic phosphate, the bond that is being broken down will release energy. How much is the value of the energy? 30.5 kilojoule. Okay. So now we're going to look at aerobic respiration. Okay, so we know when respiration there are 2 types, aerobic and anaerobic. Mana aerobic dan anaerobic. Aerobic aero, so it requires oxygen. It will use oxygen. Okay, anaerobic kalau kita tambah an dekat depan tu tidak menggunakan oksigen. Okay, I pause a moment. Ada ramai yang cuba masuk ni ke? Alright, so yeah. So we are on this slide about anaerobic respiration. So kita dah masuk 5.2 dah. So at the end of this lesson, you should be able to define what is aerobic respiration, describe briefly the 2 types of ATP production. So nak buat ATP ni sebenarnya ada 2 cara. Sama ada menggunakan enzim directly iaitu substrate level phosphorylation, and the other one is oxidative phosphorylation. Ingat tadi saya cakap tadi ada satu enzim yang sangat spesifik akan recognize enzim. Kita akan tengok sekejap lagi nama dia apa. Alright. So describe the aerobic respiration involves these 3 stages. As for today, for this session, we are trying to cover this first one, the glycolysis. Okay, so this is the overview of how you're going to break down the glucose, the process of glycolysis until the production of ATP via Krebs cycle. Okay, Krebs cycle ni kita akan tengok in next session. So basically here we are on glycolysis. So glycolysis ni kita akan break down glucose to pyruvate. So we will stop until this part. So this is the first part that is called as glycolysis. Glucose ke pyruvate. It involves 10 steps. Okay. So, and then from pyruvate converted into acetyl-CoA is called as link reaction. And from acetyl-CoA, it enters Krebs cycle and the process will continue. If this is with the adequate of oxygen, mana kalau ada oxygen, it will continue with this process. In the event of not enough oxygen, i.e. through fermentation, so pyruvate will be converted to alcohol, and there of course in the cytoplasm it will still produce a little amount of energy. So this is anaerobic. So this one with oxygen is aerobic or aerobic respiration. So yep, this is aerobic respiration, is a catabolic pathway using oxygen as the final electron acceptor. We will look onto this on next session. Okay, so mechanism of ATP generation. So mechanism nak buat, okay, we do understand that ATP, asalnya ATP ada 3 phosphate. So from 3 phosphates ni, so dalam keadaan yang energy reserve, dalam keadaan bateri yang fully charged. So macam ni nak bagi energy tu, dia akan break satu, dia akan break satu phosphate. So daripada triphosphate jadi jadi 2 phosphate only. Macam tu. Tergelik-gelik jari nak buat ni. Okay, sampai kena slash dulu. Okay, so daripada 3 phosphate jadi 2 phosphate. So daripada 2 phosphate ni, dia dalam keadaan yang low energy configuration, mana dalam keadaan yang kurang energy. So macam mana kita restore balik daripada ADP jadi ATP? That is what we're going to learn. Mechanism of ATP generation. So basically ADP kita nak campur balik dengan inorganic phosphate. Ini bukannya pi this is from what you learn in mathematics dengan sir korang tu atau madam korang tu. Okay, so it's not pi. This one I stands for inorganic phosphate. Kenapa kita panggil inorganic phosphate? That phosphate group is not bounded to any carbon compound. Okay, so ADP plus P gives you ATP. So this is the short diagram. So it is an endogenic reaction, when they can absorb energy. So the addition of phosphate group to a molecule called phosphorylation. Okay, the term that we use is phosphorylation. What does it means? Okay, phosphorylation ni maksudnya kita tambah fosfat kepada sesuatu molekul tersebut. Phosphorylation. If we remove phosphate, dia jadi nama dia, saya tambah D dekat depan ni. Sorry. Dia akan jadi macam ni. dephosphorylation. Itu kalau kita remove. Kita remove phosphate ni, kita tambah phosphate, so saya delete balik lah. So it is phosphorylation. I hope you guys get that information. Okay, so macam saya dah bagi tahu tadi, dalam learning outcome yang ini, okay, sebab tu kalau rasa saya suka in this setting saya tak present screen ni large screen, sebab I can easily jump between one slide and another slide. So here I've mentioned earlier two types of ATP production. Mari kita dah tahu ada dua cara kita nak buat balik ATP. The first one is substrate level phosphorylation. The second one yang panjang sedikit adalah oxidative phosphorylation. So kita tengok dulu yang ni. So, kenapa nama proses ini sebagai substrate level phosphorylation? Main there is a point, there is a level, dia menjadi substrate. Siapa jadi substrate? It is the ADP itself yang menjadi substrate. Dia akan bound to the enzyme. So this is the enzyme yang akan mencantumkan substrate itu, iaitu phosphoenol pyruvate ini. So dia akan ambil fosfat daripada phosphoenol pyruvate, cantum dengan ADP. So nampak dia, from ADP it will later become ATP. So the PEP, phosphoenol pyruvate, tukar menjadi produk dia iaitu pyruvate. Okay. So this process will generate ATP by transferring a high energy phosphate group from an intermediate phosphorylated metabolic compound substrate directly to ATP. So dalam human cells, this process occurs in the cytosol. Cytosol tu maknanya dekat dalam cytoplasm lah. Okay, so yang masih tertanya-tanya, apakah nama enzim dia? Okay, saya akan cakap dengan sepintas lalu. Okay, tapi saya rasa saya nak prefer you guys untuk Google sendiri, supaya korang bersusah payah sikit, so korang akan ingat. Okay, saya akan saya bagi hint lah, saya tak bagi secara penuh. Enzim kat belakang tu, nama enzim ni dia ada kaitan dengan nama dia punya substrate tu. Okay, so apakah nama enzim dia, adenosine apa? So korang cari. Nama dia panjang sikit, tak selesai ada bukan tiga suku kata, 3 phrases macam tu. Okay, cari. Okay, so nanti kalau korang dah tahu, siapa dah jumpa, boleh post dekat dalam group. Alright. So memang sekali kita tunggu-tunggu untuk korang cari sendiri info. Tak susah pun. Just Google name of enzyme for substrate level phosphorylation. Very easy. Okay, so nak bagi korang susah sikit. Alright, so and the second method. So, okay, before I jump on the second method, I hope you guys understand dekat sini what happens is that from ADP, after it bounded to this enzyme, dia menjadi ATP. And again, why it is called a substrate level phosphorylation, remember that this ADP became past tense, became the substrate. So when it is bounded to the enzyme, it has once become the substrate. Okay, so the second way, the second method, second mechanism how we can generate ATP. So ini totally tak ada kaitan, bukan tak ada kaitan, totally different daripada yang the first one. Yang first one tu very simple. Okay, so this one it involves many stages. Dia akan remove electrons daripada organic compounds, and dia akan passes electron tu akan dipass from series of electron transport chain. Main di dalam transport chain tu melibatkan banyak compound lain. So daripada one compound pergi kepada compound lain, sampailah last sekali the electron pass to the oxygen. Mana elektron tu akan sampai ke oksigen. Sebab itulah kepentingan. There is the importance of oxygen in respiration. So maknanya kita tahu oxygen as the final electron acceptor. So kenapa kita pilih, bukan kita pilih oxygen is macam tu kan. So what is the importance of oxygen ni? Dia ada kebolehan, saya gunakan bahasa pasar dulu dia ada kebolehan untuk tarik elektron tu daripada hujung chain sana sampai ke hujung chain sini. With the movement of the electron, baru dia akan generate ATP. So dia ada term yang sesuai, nanti kita akan bincang dalam next next subchapter, apakah nama term itu. Okay, so menyebabkan dia boleh sebagai final electron acceptor. Dia ada kaitan dengan redox reaction. Okay. So it involve ETC, electron transport chain, dan chemiosmosis. Ni kita kata on the next subchapter, contributes to the most of ATP production. Mana paling banyak ATP diproduce dengan cara ini. Okay. Alright, so inilah gambaran secara ringkas. Macam saya dah bagi tahu tadi, mana oksigen dalam gambar ni? Saya tak nampak oksigen lah pula. Oh, ni here. Okay, so actually it's not big enough. Okay, so what I'm trying to tell you just now is, can you see this yellow arrow moving from here, from one compound to another compound complex, from one complex to another complex, blah blah blah, until finally. Okay, this yellow arrow depicts, showing the movement of the electron. So electron ni last kali dia bergabung dengan oksigen ni menjadi H2O, water. So it comes by no surprise. That's why when you exhale ni hembus nafas tu, bila kita kurang ha dekat cermin tu kan, ada water molecule. So antara datangnya daripada sini jugalah. Okay. So this is the redox reaction. Surely you have learned this many times and you guys have mastered this in your chemistry classes. So if you haven't mastered this in your chemistry classes, so there's something wrong with your chemistry, and there's surely something wrong with our chemistry as well. So, but let me help you one more time again. So when we regard as redox reaction, it involves oxidation and reduction. So I've added this mnemonics for this slide, but it is not in your slide. It is on my slide here. Add extra points over here. So oxidation is loss of electrons or hydrogen ion. So OIL. Can you see this? OIL. So jadi oil. Okay. Reduction is gain of electrons or hydrogen ion. Oil rig. So RIG. Oil rig ni kan pelantar minyak. So I always use this term pelantar minyak, oil rig. So I always remember myself. I always tell myself oxidation is loss, because sometimes we get confused. Siapa yang lost electron? Siapa yang terima elektron? Siapa yang lost hydrogen ion? Siapa yang terima hydrogen ion? Siapa yang tengah oksida? Siapa tengah reduce? Always remember this. Siapa yang hilang elektron, dialah yang mengalami oxidation. Siapa yang, okay, contohnya saya tunjuk dekat sini kan, dah senang ada gambar kat sini. So nampak so remove 2H ion. So mana lactic acid mengalami, saya gunakan bahasa Melayu dulu. Lactic acid mengalami oxidation menjadi pyruvate acid. So tengok dekat sini, dia lost kan, dia hilang hydrogen ion kan. So dia oxidize lah, oxidation. Yang ni dia reduction sebab dia gain, dia add, tambah 2H. Gitu, tak susah pun. Okay, so this is structure of NAD+. Ni nanti korang akan tengoklah structure NAD+ ni dekat dalam ETC. Sebelum ETC pun korang belajar dalam Krebs cycle juga, korang akan belajar ni. Okay, apa itu Krebs cycle? Jangan risau, there's not for what you're going to worry at this session. That will what you're going to worry on the next session. Okay. So the redox session always use this NAD+. Okay, so you can see here, where do I start? All right. So okay, yeah, for NAD plus gains hydrogen ion dan elektron ni, so dia being bukannya dia gain, dia hilang. So reduction of NAD plus, bila kata reduction, dia gain lah, nampak dia dapat dia plus. Reduction is, nampak, even saya salah guna pun saya boleh bukan terkeliru, tersasul. But if you do a self check, you will be in a good hand. How do you do a self check? Very easy. Remember this, oil rig. Nampak saya buat show last sikit lah. Saya tambahkan. And most dekat banyak step saya tambah dekat atas ni. Always to remind you and remind myself as well. So NAD plus is reduced to become NADH. Gitu ayat dia. So kenapa kita tahu dia reduce? Sebab dia gain, dia dapat hydrogen ion dan dia juga dapat elektron kat situ. So this one I'm going to skip this slide sebab dia sama dengan apa yang saya dah elaborate tadi. Okay, so the big deal about aerobic respiration ni secara penuhnya, inilah gambarannya. So we have series of events atau series of processes. Kita akan mula dengan glycolysis, Lepas tu kita sambung dengan Krebs cycle. Last but not least barulah ETC. But Krebs cycle and ETC will be discussed on our next session. Okay, for this session kita hanya bincang pasal glycolysis sahaja dulu, insyaAllah. I.e. how do we convert glucose to become pyruvate? Ya, glukos yang korang makan yang ada dekat dalam minuman berkarbonat korang, yang ada dekat dalam mana-mana Pizza Hut korang, dekat dalam fries, semua tu lah yang dalam carbs, nasi yang korang makan, breakfast korang makan pagi tadi, ada glucose dalam tu. So that is what we are going to deal with, that's what we're going to discuss. How do we convert that glucose into pyruvate? But if you still wonder, semana datang glucose ni, daripada air je ke? Tak, kan kita dah tahu dia di breakdown by the by your digestion process. Okay. So this diagram is also very good in terms of, can you see, later you're going to need this, you're going to refer to this diagram many times after this, because you can see through glycolysis, we know at the end of glycolysis, it will produce 2 ATP. From link reaction, another ATP. So through this, another ATP. Link reaction through ATP. Krebs cycle or citric acid cycle produce another 2 ATP. So you're going to refer to this diagram many times after this. All right. Okay, so we have come to our big deal of our session for today. Okay, glycolysis. So namanya pun glycolysis. Glyco stands for glucose, lysis stands for breakdown. So kita nak pecahkan glucose, nak potong dia. But how? Okay, so before that, this process takes place in the cytosol. Okay, dekat dalam sel. And first stage, this is the first stage of both either aerobic and anaerobic respiration. Maknanya aerobic dan anaerobic respiration mesti dia akan start dengan glucose dulu. So inilah persamaan antara kedua-dua proses ni. So cumanya apa yang jadi pada pyruvate itu with and without the presence of oxygen, itu yang akan determine sama ada dia menjadi aerobic atau anaerobic. So one molecule of glucose is split by a series. Bila kita kata by a series tu maknanya step by step, bukan yang sekali split terus dah siap jadi. Tak, step by step. Okay, series of reactions into 2 molecules of pyruvate. So here let me here I want to get this straight with you guys. So from one glucose dia akan hasilkan 2 pyruvate. Okay, so logic dia very simple. We know glucose have 6 carbons. Okay, that's why dia C6H12O6. C6 ada 6 carbon. So at the end of the reaction, you akan hasilkan 2 pyruvates where each of the pyruvate have 3 carbons. So logic lah, daripada 6 carbon, macam mana 6? 6 carbon, you split into 2 3 carbon compound. Okay, logic kan? Okay, so later you get to see how this happen. Alright. What else do I need to add from here? So 2 pyruvate over here, blah blah. Okay, it's very simple as that. Tak ada apa-apa pun yang complicated sangat. Yang complicated ni is how. Alright. So glycolysis comes in 2 phases. The first phase is the energy investment phase. The second phase is energy payoff phase. Ada fasa invest, ada pula fasa payoff. Let us look at this diagram. Okay. I'm clearing my throat, not purposely, bukannya sebab ni saya nak bagi korang highlighted up sikit. One, so as I mentioned earlier, there are 10 steps involved in this conversion of glucose to become pyruvates. And shall I say pyruvates with S? Because nanti ada 2 pyruvates. Pyruvates with S bukannya nama dia pyruvates. Pyruvates with S tu grammar ada 2, lebih pada 1 kan. Okay. So the first 5 steps steps is the energy investment. So we first have glucose and then it converted into glucose 6-phosphate and then converted into isomerized into fructose 6-phosphate and then it further phosphorylated to become tengok saya gunakan term-term tu lah. Saya nak biasakan korang dengar term-term sebab kenapa saya cakap phosphorylated tambah phosphate gitu. So become fructose 1,6 bisphosphate, bukannya bi bukannya bi just bi saja, ada s kat situ. Bisphosphate and then from fructose 1,6 bisphosphate, and you split them into two compound, which is the first one dihydroxyacetone phosphate, and the other one is glyceraldehyde 3-phosphate. Full stop until there. That is the first energy investment phase. Apa itu energy investment phase? Okay, investment ni macam korang belajar macam dalam ekonomi or kimia or mathematics and so on. Invest maknanya kita kena laburkan dulu. Labur maknanya kita kena macam masukkan modal dulu lah. So what is the modal untuk pelaburan ini? First, at this phase we are using 2 ATPs. Okay, so here at step number 1 and at step number 3. So ada 2 kali ATP tu dipakai. So maknanya kita dah negatif lah. Kita dah pendek cerita gila. Saya dah laburkan RM1,000 dekat sini at step number 1. Saya laburkan lagi another RM1,000 at step number 3. Okay, and then at energy payoff phase, okay, so from glyceraldehyde 3-phosphate, later it will be converted into 1,3-bisphosphoglycerate. I mean, it's another phosphorylation again. But bila saya kata, okay, name of the process is phosphorylation, tambah phosphate. Name of the event, mana kalau kita tukar dia kepada language, phosphorylated, dia setelah diphosphorylationkan. Gitu, we just playing with language over here. So at step number 6 ni, glyceraldehyde 3-phosphate ni phosphorylated to become 1,3-bisphosphoglycerate. About the numbering naming nanti saya akan explain later in detail. Okay, so and then the next step, from 1,3-bisphosphoglycerate dia akan menjadi 3-phosphoglycerate. So what I want you to notice here, dia asalnya ada 2 phosphate, now dia jadi 1 phosphate sahaja. That phosphate was be given to, dibagi kepada ADP. ADP ni jadi ATP. See here, kita hasilkan 1 ATP. Dah dapat RM1,000 dah dekat sini. Okay, as we go down the process at step number 10, the final step, another 1 more ATP is being produced. Okay, so dapat another RM1,000 kat sini. Ingat tadi asal-asalnya kita dah laburkan RM2,000, and at this step kita dah dapat RM2,000, tapi belum lagi untung. So apa untungnya kalau saya laburkan RM2,000, saya dapat RM2,000. Baik saya tak melaburkan, baik saya tidur, atau kata memang kalau tidur je pun. Okay, but remember, the splitting of fructose 1,6 bisphosphate ni dia bagi 2 3 carbon compound, DHAP and also G3P. So 1 G3P hasilkan 2 ATP. So can you see this back and forth arrow ni? Maksudnya DHAP, dihydroxyacetone phosphate ni, also can be isomerized, converted to become glyceraldehyde 3-phosphate. So when it become glyceraldehyde 3-phosphate, it will go through the same process until it become pyruvate. So it also generates another 2 ATP. So in total, during energy payoff phase ini, you akan hasilkan 4 ATP. So you akan dapat RM4,000. Gitulah. So you laburkan RM2,000, you dapat RM4,000. So what is your total what is your total gain of this first glycolysis ni? You dapat 2,000. I mean 2 ATP lah. Mata duitan betul 2,000 je celik, 2,000 je celik. Kau cakap 20% kau tak celik. So and also actually in this diagram you boleh nampak lah dia ada tulis nombor 2 sebenarnya kat sini. If I enlarge this, ya, kalau kau tak nampak dengan mata, tengok mata hati. Tak nampak mata hati, tutup je lah. So 2, kau nampak ni 2 ATP, 2 of these, 2. So nombor 2 ni bukan yang tambahan, bukan sahaja dilukis kat sini. It means ada 2 ketul, I mean 2 molekul. How can you have to remember 1 G3P yang DHAP tukar kepada G3P kan jadilah 2. Gitu. So you're also going to need 2 NAD plus to become 2 NADH, 2 ADP becomes 2 ATP, and the process continue. Gitu. Okay. So I'm going to go down into detail for each of the phases. So I have roughly about 10 minutes. No, I have more than 10 minutes. Actually, I have 20 minutes. Okay. So I'm cracking all the bones. Let us get started with this. Okay. Yep. So we begin with the energy investment phase. Tapi saya tak risau sangat sebab saya dah cakap hampir semua dah tadi. Okay. So investment phase ni, ATP provides activation energy by phosphorylating glucose. So kat sini apa yang saya emphasize sikit lah nama-nama terms dia. Nama-nama proses, okay. Apa tukar kepada apa. Alright. So this process requires 2 ATP per glucose. Dekat fasa investment ni lah saya dah sebutkan tadi pun. So ATP is produced by substrate level phosphorylation. Ya, yeah. ATP ni dihasilkan dengan cara substrate level phosphorylation. Maknanya memerlukan enzim. Okay. NAD plus is reduced to NADH plus H. And 4 ATP and 2 NADH plus 2H are produced per glucose. So ini adalah final calculation dia. Okay. So saya dah tampalkan juga kat sini oil rig ni kat atas ni for your reference. Okay. So ini adalah summary. Energy investment buat apa? Energy payoff buat apa? Payoff tu yang bawah ni. Yang bawah ni. Okay. So if you confuse bawah mana sih? Ni is same with this one, bawah ini. So actually dekat sini kan dia ada macam arrow atau macam bow arrow, macam anak panah gitu kan. And start something here. From this one until this one, this 6th step until 10th step is the payoff, which is this one. Okay, so now we're going to each step of the energy investment phase. Okay, so we look on to the first one. Okay, step number 1. Sekejap, saya serahkan kipas tu. Okay, step number 1. First, we convert glucose to become glucose 6-phosphate. So maksud saya arrow kat sini lah. Glucose converted to glucose 6-phosphate. And the enzyme, okay, sebelum tu saya minta maaf awal lah. Yeah, if you're going to ask me, sir, kita orang kena hafal ke nama setiap benda-benda yang pelik-pelik ni? The answer is yes. So that is why later I'm going to require you guys to do on your own self practice. Keep on memorizing these names over here, but I'm going to give you tips later on. Okay, so yeah, memang korang kena hafal. So korang kena cubalah kaedah yang untuk hafal dan dia. Tapi ada cara dia. So yang ni saya beritahu first step. Glucose tambah 1 fosfat jadi glucose 6-phosphate. Kenapa nama dia glucose 6-phosphate? Because you are having the phosphate at carbon number 6. Sir, how do you know that is carbon number 6? Okay, so that's very easy because if you refer to the structure of glucose, so this is structure of glucose. Rasanya aku dah ajar korang dah last time ni kan. Sekejap saya besarkan sikit. Sekejap saya besar. Tak gerak pula. Degil ni. Macam korang je. Okay. Alright. So we have carbon number 1 over here of the glucose, carbon number 2. Okay, please refer to the arrow. Kan saya punya cursor ni ada 4 arrow kan. Refer arrow yang atas. Kan ada cabang kiri, cabang kanan, cabang atas. Refer to arrow yang atas. So this is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5 over here still inside the ring. And where is carbon number 6? It is outside of the ring. It is above of the ring. Gitu. So, dekat sinilah nanti dia akan tambah phosphate tu. Sebab tu dia tambah jadi kat carbon number 6, bukan dekat carbon number 1 or any other carbon. Dia tiba-tiba, saya boleh tak? Kita fancy sikit lah, kita tambah kat carbon number 4. Suka tingkah nak membongak, tak boleh. It's not you for you to decide. Okay, dia memang dah kejadian dia macam tu. Okay, so and name of the enzyme, sorry memang korang kena hafal. Okay, so macam mana hafal nama enzyme ni? Hexo tu kan hexagon, bukan hexagon tu kan 6 segi. It is referring to this 6 carbon compound. Okay, so phosphate group is transferred from ATP. Nampak phosphate yang tadi daripada mana? Daripada ATP. Okay, to glucose. To bind to the 6th carbon. So glucose 6-phosphate is more reactive. Alright. Okay, so that is the first step. Second step is isomerization. Okay, so isomerization, as what we have learned in chemistry as well, here membuat isomer. So there's nothing change over here. There is no addition of something. There is no removal of something. It is just you rearrange the molecules. Okay. So what is the change here? If looking at this simple diagram over here, you can't see any difference because it is still 6 carbon having the glucose at having the phosphate at carbon number 6, still semua sama je. But what is the difference? Okay, kita tengok sekejap lagi. Glucose 6-phosphate is rearranged, okay, ataupun saya akan gunakan term isomerized to its isomer fructose 6-phosphate. Tengok dekat sini. Okay, so this is glucose 6-phosphate and this is fructose 6-phosphate. Look, I mean, please pay attention to the shape of the ring. Um, so glucose 6-phosphate is having a hexagon shaped ring. Hexagon lah bentuk segi 6. Whereas fructose is having pentagon shape, berbentuk 5 segi. Segi 5, 5 segi. Apa-apalah ikut suka hati korang lah. Kan? So why does this glucose need to be isomerized to become fructose? Kau tengok sini. Here is the interesting part. Remember, bear in mind, glucose ni nanti kita nak potong dia jadi dua. Sebelum kita nak potong jadi dua, mesti kita nak cuba balancekan dia lebih kurang sama kiri dan kanannya. Baru boleh potong dua. How do we balance this? Okay, but this is not a technical, actual technical explanation lah. This is just demographic. This is just my self explanation gitu. So tengok kat sini. We already had the carbon number 6 outside of the ring, main di luar daripada ring. So we are going to do the same for carbon number 1. So we have rearranged the carbon number 1 to be outside of the ring as well. So nampak dia terjulur keluar daripada ring. So the actual ring is this one lah, yang 5 segini. Barulah hampir balance. So why is not balanced yet? There is something wrong because at this left side of the fructose, namanya pun fructose 6-phosphate, you only have phosphate at carbon number 6. What about carbon number 1? Tak sama, tak adil lah kan. So you must add also another phosphate at carbon number 1, makes it even left and right. So kau nampak ni, kiri kanan tu sama. Barulah adil kan. Okay, so how do you got this? Of course lah, again, ambil lah daripada ATP lagi. Siapa lagi nak bagi dekat dia kan? And again, to my pardon, sorry, you have to memorize the name of the enzyme, which is in this case phosphofructokinase. Okay, tak susah pun. Fosfor tu because kita nak tambah fosfat. Frukto, kenapa frukto? Dah sebelum tu fructose. Kau nak bagi nama apa lagi? Kinase. Sebab kinase ni kan ingat ada types of enzyme. Kinase ni yang pindah-pindahkan functional group. So what is the functional group? Is the phosphate. Okay. Yang tadi tu yang isomerize kan tadi tu, nama enzimnya adalah phosphoglucoisomerase. Isomerase tu kan isomerase, nama enzim isomerase kan. So dia nak tukar-tukar fosfor sebabkan dia tukar-tukar apa ni, fosfat, glucose 6-phosphate, macam itulah. Okay, so tak susah sangat pun. So tengok sekejap kita dah cover sampai step number 4. Number 3. So saya check masa, kita dah 10 minit lagi. Wait, where am I just now? Tadi baru first. Okay, so I'm here. So isomerization dah senang settle. So phosphorylation, yang saya baru just cakap tadi, mana kita tambah another phosphate to carbon number 1. Sebab tu namanya fructose dash 1, dash bisphosphate. Sebab kenapa ada 1 dengan 6 tu? Because the phosphate is at carbon number 1 and carbon number 6. Semudah itu. Tak susah-susah. Bio ni tak susah. Yang susah tu maths, chemistry, physics, tu semua susah cegra je. Bio ni ingat senang je. Fructose 6-phosphate, okay, tengok akhir dekat sini. Dia gunakan arrow. So my sentence would be fructose 6-phosphate, sebenarnya ada pun kat bawah ni, phosphorylated to, dia tu phosphorylated jadi apa? Fructose 1,6 bisphosphate. Okay, here is my tips for you. If you are tak sure, saya tak sure lah apa nama proses ni kan, saya hentam salah kan. Betul-betul kalau kau hentam tu dah likely tu salah. So to save yourself, the way how we mark students answer is by we reading sentence per sentence. I mean, if that sentence all have correct content, semua betul, dia betul. Tapi kalau ada contradiction, contohnya fructose 6-phosphate dephosphorylated to become fructose 1,6 bisphosphate. Apa tukarkan apa dah betul, tapi term kat tengah tu tak betul. Biasalah manusia macam korang kan, buat salah, buat masalah tu lain. So maybe if you are not sure, if maybe if not pro enough, so you can separate, you can choose the safe side of this problem. So what you can do, you can always just play safe. Just cakap, you need to break down the sentence into two sentences. Fructose 6 phosphate converted. Apa-apa pun kau tukar je converted. Kau cakap je converted. Ataupun yang perkataan sewaktu dengannya lah. Converted to become fructose 1,6 bis phosphate. The next sentence baru you cakap fructose 6 phosphate is phosphorylated oleh siapa? Oleh the enzyme phosphofructokinase. Kalau kau tak yakin juga nama enzim tu pun apa, breakdown kepada is phosphorylated sahaja dah cukup. Mention nama enzim dalam ayat lain. Sebab kita nak cuba elakkan permasalahan korang yang besar ni. Okay. Okay, why do I need to mention you guys this? Sometimes this simple part of the question, part of this explanation carries a lot of mark. Maybe it carries 2 marks over there. Where the marks comes from, the marks will be avoided when you mention apa tukar jadi apa, nama proses ataupun nama enzim. So nampak dia boleh breakdown kepada 2 markah kat situ. So kalau dah campur adukkan 2 markah yang salah dalam satu ayat, dua-dua tak dapat. Begitu. Alright. So ayat tambahan lagi, point extra lagi. Phosphate group is transferred from ATP to fructose 6 phosphate. So dekat situ. Okay, next. Step number 4. Splitting of 6 carbon sugar. So tadi dah fructose 1,6 bis phosphate kan. Dah ngam, dah balance, dah kiri kanan ni. Dah ada phosphate, single phosphate. Baru kita nak potong tengah dia dua-dua. So nak potong pakai apa? Fructose 1,6 bis phosphate, okay, is converted to become glyceraldehyde 3-phosphate, G3P yang ini. Dan jangan lupa, dihydroxyacetone phosphate, DHAP. Okay. So fructose 1,6 bisphosphate is unstable yang ini, so it will split into two different 3 carbon sugars, G3P dan DHAP. Okay. Next, step number 5, isomerization lagi. So DHAP is isomerized to become glyceraldehyde 3-phosphate. Ataupun kalau korang tak sure, boleh juga break down the sentence. DHAP converted to G3P, full stop. Dan cakap lagi, the process is isomerization. Begitulah. Okay, ataupun this step is called as isomerization pun boleh. So DHAP rearranges into G3P. Okay, so one glucose molecule will produce 2 molecules of G3P. Sebab yang satu dah split tu dah jadi G3P, DHAP pun tukar jadi G3P. So G3P jadi apa? Jadi 2. Okay. G3P is used as a substrate for the next reaction. Tengok, ini adalah next reactionnya. So, tadi saya nak tambah lagi nama enzim yang saya tertinggal. Untuk split, untuk potong fructose 1,6 bisphosphate tu kita gunakan aldolase. Okay. Nak isomerase kan? Of course kita guna isomerase. Okay, we almost have come to the end of glycolysis. Okay, so tadi tu yang panjang-panjang kita cerita, the first 5 step itu until here ni, ini adalah energy investment phase. Kenapa invest? Kita pakai ATP sekali kat sini, kita pakai lagi sekali dekat sini. Dah 2 kali kita pakai. Okay, so now barulah fasa untuk bayar semula atau mendapatkan balik keuntungan kita yang juta-juta tu. Okay. So, step number 6, we start with oxidation and phosphorylation. Do bear in mind, please notice, please pay attention here. Ada 2 event, ada 2 perkara, ada 2 proses yang berlaku dekat sini. Bukannya hanya satu sahaja, ada 2. Okay, so basically, glyceraldehyde 3-phosphate, i.e. G3P, converted to 1,3 bisphosphoglycerate. So itulah nama term yang kita pakai, 1,3 bisphosphoglycerate. Remember again, it is bis, B-I-S, bukannya by. I know there are some references, buku cap ayam, buku cap kucing, buku cap harimau semua tu, yang tak tahulah brand apa pun semua tu, dia kadang-kadang ada gunakan nama pelik-pelik. Ada yang gunakan DI, ada yang gunakan BI sahaja. I mean, biphosphate lah, diphosphate. No. We use this one, we use this term, we follow Campbell. Okay. Each glyceraldehyde 3-phosphate is oxidized. Nampak? Now we are using this term, oxidized. Okay, so tadi tu untuk markah pertama untuk selamatkan diri, kita cakap apa tukar kod apa. Markah kedua kita cakap markah berapa. So korang tanya, soalan keluar dalam PSPM ke? Apa-apa soalan pun boleh keluar. Ya. Okay, yang tak keluar muka saya guys tu. Memarahi korang yang tak betul-betul. Okay, so each G3P is oxidized by the transfer of H atom to NAD+. Okay, saya mudahkan kalau kita kat sini kan. G3P oxidized by the transfer of hydrogen atom to NAD+, forming ataupun to become NADH plus H plus. Plus H plus tu campur hydrogen ion, bukannya plus H plus apa lagi, plus hydrogen ion. Okay, each G3P ni combine with inorganic, tadi tu satu event, satu part of the story. Another each G3P combines with inorganic phosphate. So kita kena emphasize kat sini inorganic phosphate. Kenapa nak emphasize inorganic phosphate? Maknanya kita nak cakap, kita tahu phosphate ini bukannya datang daripada mana-mana ATP. Dia memang ATP. Memang phosphate yang berenang-renang ke tepian, yang free moving, free streaming phosphate dekat dalam cytosol itu. Okay, so berlaku kat mana? In the cytoplasm to form 1,3-bisphosphoglycerate. So glycerate. So dia akan bind kepada first carbon. Yang asalnya dia dah ada carbon ni dekat carbon. Dia ada phosphate ni dekat carbon number 3. Sebab tu namanya glyceraldehyde 3-phosphate. Now namanya 1,3-bisphosphoglycerate sebab dia ada phosphate dekat carbon number 1 and dekat carbon number 3. So again, apa oksidasi Lioger ni? Abaikanlah. So kita pakai oil rig. Saya tak suka ini. Sekejap. Saya suka atas tu. Oil rig. How many minutes? Saya ada dalam 2-3 minit lagi. So insyaAllah sempat. Okay, step number 7. Substrate level phosphorylation. Yang ini pun sama juga. Bila kita kata substrate level phosphorylation, dia involve enzyme lah. So 1,3-bisphosphoglycerate converted to 3,phosphoglycerate. Okay, 1,3-bisphosphoglycerate transfer 1 phosphate group kepada ADP. Bila dia bagi ke ADP, ADP jadi apa? ADP jadi ATP lah. Nak jadi apa lagi? ATP are formed through substrate level phosphorylation. Formation of 2 ATP pay off the original energy investment. Okay. So kita phosphate yang ni dia bagi dekat ADP jadi ATP. So dia jadi apa? 3-phosphoglycerate. Okay, kalau korang perasan, ini dia tak sama dengan ini. Dia tak sama dengan glyceraldehyde 3-phosphate. Ini namanya adalah silap. Ini adalah namanya 3-phosphoglycerate. Kenapa ada perbezaan antara selepas step number 7 dengan before step number 6 ni? Remember, sebab sini ada oxidation yang berlaku. So dia ada dia ada kehilangan hidrogen dekat situ. So sebab tu G3P tidak sama dengan 3-phosphoglycerate sebab dia dah ada kehilangan hidrogen tadi masa kat step number 6. Okay, so and then berlaku isomerization. Senang je, mudah. 3-phosphoglycerate kata asyik phosphate ni kat carbon number 3 menyemak kita buka tengah pula kat carbon number 2. Okay, so jadi now 3-phosphoglycerate converted to kepada 2-phosphoglycerate. So 3-phosphoglycerate is isomerized to become 2-phosphoglycerate. So please use the name of the process. Okay, so second last, kita nak habis dah pun, jangan risau. 2-phosphoglycerate, okay, dia mengalami dehydration. So dehydration ni maksud apa? Kehilangan air. Okay, hydration kalau kita tambah air, dehydration kita buang air. So removal of water. So the term ayat kita gunakan 2-phosphoglycerate is converted to phosphoenol pyruvate. 2-phosphoenolpyruvate mengalami dehydration to remove water molecule. Okay, so dia juga rearrange lah ke bawah sini. And last but not least, another substrate level phosphorylation di sini, phosphoenolpyruvate dia converted to become pyruvate. Each PEP, phosphoenolpyruvate ni, transfer phosphate group kepada ADP. So ADP tukar menjadi ATP. So ada berapa ATP? Ada 2 ATP. So net yield of energy from glycolysis adalah 2 ATP for each glucose molecule yang saya dah explain tadi. So this is the diagram showing the step. Okay. So for this one, nama enzyme yang kena hafal last sekali ni sahaja. Pyruvate kinase. Itu yang biasa ditanya lah. Okay. So End product of glycolysis ni adalah summary dia. Okay, so dari NADH ada 2 ATP sebab 4 ATP terhadap 2 ATP jadi 2 ATP, and also 2 pyruvate. Jangan lupa pyruvate yang dekat last sekali tu. So that is glycolysis. So you start with 6 carbon compound, hasilkan 2 ATP, hasilkan 2 NADH, and also 2 pyruvate. Alright, so I think that is for today. Ngam-ngam, ada bukan ngam-ngam, saya advance by 4 minutes lagi pun. Okay, so ada soalan kat tadi? Masih survive lagi? Masih hidup lagi? Okay, I know it is not an easy process for you to digest quickly, but I hope you guys can look through this video one more time again or later, so that you guys can learn better, inshallah. Okay, so I will end the recording session first. Okay.